Today we are covering the last piece of our fact paper. We've spent a few lessons going through the experiment in question 1, and now we are looking at how we can use the microscope work in question 2 to actually work as revision, even if we don't have a slide. Okay, so we're told that J1 is a slide of a stained transverse section through a plant leaf. You're not expected to be familiar with the specimen. They could give you absolutely anything. The idea is that you can follow instructions and that you're going to see general features that are similar between different specimens. Okay, so the specimen on J1 has an irregular shape. Okay, so it's not looking like our standard plant leaf in our textbook, all right? Then they remind us, before doing anything else, use a sharp pencil for drawings. Now, it's really important that you follow that instruction, because if your lines are too fat, you can actually lose marks for your drawing. So make sure that you've got a very sharp pencil for your drawing. Then they ask you to draw a large plan diagram of the section of the leaf shown by the shaded area in figure 2.1. Use one ruled label line and label to identify the vascular bundle. So that is the only thing that we are going to label. One line, one label, one vascular bundle. Finished. Okay, so we have an upper and lower surface. We see there's a bump in the middle and we're asked to draw this section. Okay, you are expected to draw the correct shape and proportions of the different tissues. Again, standard instruction, but a nice reminder for you. So obviously, if you don't have J1, you don't know what J1 looks like, but you can get a good practice still doing a plan diagram if you use your textbook. So for example, on page 130, there is an image, a photomicrograph. So you've got an image of a leaf and you have a similar bump in the middle. So we can draw the relevant piece out of that image instead of the textbook. Okay, plan diagrams, remember, don't have any cells whatsoever at all. We're just drawing boundaries of the tissue. So if we work with that image from the textbook, we would end up with something like, the top also dense on this one. Remember, you always draw what you see. We've got a very thin epidermal layer at the top. Okay, you would be doing this in your very sharp pencil. Okay, we don't draw an end off here because it carries on. So we let it carry on the sides. Okay, then in the middle, we have a bit of a bobble. And then from here, we go across again. So there's a whole lot of cells in each of these zones. Okay, there's your palisade cell zones. So you can see them, longer, taller, thinner. Okay, column-shaped cells. Okay, but we're not drawing each of those cells. We are just drawing the lines where the different cell type changes, because that's the edge of a tissue. So a plan diagram is always about the tissues. So then we come and we have another sort of scoop here. And around that, there's another scoop. And around that, if we look carefully, down here, there's another scoop. Okay. Then from about here, we go off with our spongy mesophile in the blade of the leaf. And the same thing on this side, though, there's some more fun and games happening here. We've got another vascular bundle here in the specimen that we're drawing, okay? Dodgy, dodgy, bad. Okay, lines must be smooth, they must meet perfectly. No tails, no overlaps, no featherings. Okay, and then we have our lower epidermis. So we have a big scoop here. Not quite like that though. Remember, we want to try and be accurate. So more to a point and then up. And then following this side. And again, that epidermal layer, we're not going to draw each of the cells, we're just drawing a second line to mark the other edge of the tissue. So very thin line all the way around. Okay, so it's not the J1 that's in the question, but it's a good practice for drawing a plan diagram. And we can still do the practice of the labeling. They want one line, one vascular bundle. So there's our one line and our vascular bundle. Finished. And we leave it. We don't go labeling other things. We never shade anything. Okay, it's all just lines to indicate the edges of a tissue. And we move on. Observe the upper epidermis and palisade layer in the leaf. Okay, so again, we're looking at J1. Select one group of 2 adjacent touching cells from the upper epidermis and 1 adjacent touching cell from the palisade tissue below. Each cell must touch at least one of the other cells. That's really important to find these touching cells, because if your cells in your diagram aren't touching cells, then you haven't followed the instructions in the question. So when they say one group, 3 cells, 2 of this, 1 of that, all touching, you must follow any instructions that they give you like that, okay? Make a large drawing of this group of 3 cells. Use one ruled label line and label to identify the cell wall of 1 cell. See how they put things in bold, okay? That's to help you. It's to make sure you follow these instructions. Okay, so we're going to use that same photomicrograph from page 130. Obviously, we can't zoom in the same way that we would zoom in on our microscope in our prac, because we wouldn't want to draw our plan diagram on a low power objective, and then we want to zoom in on a high power objective to do this detailed diagram. But we can get an idea at least of the relative sizes and orientations of these cells that we're drawing from page 130. So, epidermal cells. Okay, again, beautifully neat, tidy. Okay, and they are touching. Okay, but they have cell walls, so we need a double line around each cell. And there we go. So maybe on the J1 slide, when you zoom in, maybe you can see a nucleus, for example. But in the cells that we're drawing from page 130, we can't see that kind of detail. Okay, one palisade cell, he would look something like this underneath. And it's important also that you fill that entire space that you're given to draw in. And again, palisade cell, plant cell, cell wall, 2 lines around the outside. So maybe in J1 we have a really thick cell wall on the epidermal cells and not so thick on the palisade cell. There's all sorts of different things where we can see variation in what's actually on the slide. But there's nothing wrong with getting this practice working off images. Okay, 1 label line, 1 label, 1 cell wall. So let's do that one. And there he is, cell wall, done. Okay, a little bit quicker when we draw them like this, rather than drawing them from a microscope. Because when you're working on the microscope, you do want to take that time to be precise, to make sure you know what you're looking at, to find a good 3 cells to draw. Okay, and all of that takes a bit of time. Then you want to draw far more accurately and neatly than I have done here. But of course, a pencil on a paper is quite different from a digital pen on a pad. Okay, so let us move on. Observe the leaf on J1. Eish, another again. Okay, so suggest one observable feature of J1 which supports the conclusion that this plant grows in a dry environment. Explain how this feature would prevent water loss. So obviously we can't see J1. So we do, however, know quite a bit about zero-fitting adaptations. So we can go ahead and pick one. We can think about that whole long list of adaptations and what would we like to see on J1. Okay, so a feature could be, very common feature, would be sunken stomata. And how does that prevent water loss? Well, we're trapping that moisture near to the stomata, so we're reducing that gradient of water, so we're reducing diffusion. So traps moisture near to stomata reducing diffusion out of the leaf. Okay, so we're going through this question a lot faster than we went through the experimental question. There's a lot more theory when it comes to these. Okay, so figure 2.2 shows a diagram of a stage micrometer scale that is being used to calibrate an eyepiece graticule. One division on either the stage micrometer scale or the eyepiece graticule is the distance between two adjacent lines. The length of one division on this stage micrometer is 0.1 millimeter. Okay, so which one is your stage micrometer and which one is your eyepiece graticule? This big fat one, where you can't see what's going on, that is your stage micrometer. It's the one that's sitting on your microscope slide. Okay, this one here, where you can see the full scale and it fits nicely and it goes across, there is your eyepiece graticule. Okay, so one division on your stage micrometer is all of that. And we even have a length here. This is 0.1 millimeters. One division on an eyepiece graticule is one of these tiny little ones. That's, remember, because it's between two adjacent lines. So here we have 10 divisions between 0 and 10. How nice the numbers line up for us. Okay, so they want us then to do something with this, I'm sure. Complete figure 2.3 by using figure 2.2 to calculate the actual length of one eyepiece graticule unit. So at this magnification, because remember, we can only calculate the length of an eyepiece graticule unit as the specific magnification. As soon as we change magnification, the length is going to change. So here's figure 2.3. One eyepiece graticule unit equals this divided by that equals so many millimeters. Okay, so we're going to need a millimeter measurement. Okay, well, there's our 0.1 millimeters. So 0.1 millimeters divided by how many EGUs fit into that 0.1 millimeters. Well, we're going from 10 to 50. So 50 minus 10 is 40. So there are 40 EGUs in one stage micrometer. So one of a 0.1 millimeter divided by 40 will give us a 0.0025 millimeters. So each of these single little tiny divisions is 0.0025 millimeters. It's tiny. Okay, but step 2. Convert the answer to a measurement with a unit most suitable for use in light microscopy. Well, this one, of course, is our micrometers because it's a light microscope. So we're aiming for micrometers. So we would take, to convert from millimeters to micrometers, we would take our 0.0025, we would multiply it by 1,000, and that would give us 2.5 micrometers. We really want to be sure that we put that unit in because that is our most suitable unit. We be very sure whenever we're changing units that we put them in. So we actually should have 0.0025 millimeters multiplied by 1,000. That's even better. So now we know at this level of magnification, one of our EGUs, our eyepiece graticule units, is equal to 2.5 micrometers. So what next? Figure 2.4 shows a photomicrograph of a transverse section through a leaf taken using the same microscope with the same lenses as figure 2.2. So magnification is unchanged. Microscope is unchanged. The calibration that we have done, we can now use. So you are not expected to be familiar with this specimen. What's quite funky is this is actually a specimen from a previous exam year. This is one of the slides that they used for an October-November exam one year. But don't ask me which year at the moment. Okay, so anyway, use the calibration of the eyepiece graphical unit from B part 1 and figure 2.4 to calculate the actual length of the plant leaf from X to Y. Show all the steps in your calculation and use appropriate units. Now, you've said that the appropriate units for light microscopy are micrometers. So that's what you're going to need to work at. So what are we looking at? We are looking at here from X to Y. Well, Y is nice and easy. That's pretty much bang on 70, right? X, we're going to have to look a little bit squigglier. We're looking here, let's say that is 26. So from 70 to 26 is the width of our leaf. The length of the plant leaf from X to Y, not the width, because the width is going to change. It's a funny shaped plant leaf, this one. It's a whole actual triangle. Okay, so how many EGUs have we got? We have 70 minus 26 equals 44 eyepiece graticule units. We, however, know, because we've calculated, that one eyepiece graticule unit equals 2.5 micrometers. So we can go back here and we can say, well, if we've got 1 is 2.5, then 44 is 44 times 2.5 of them, which is going to give us 110 micrometers. Units again, important. So let's be biologists, we'll put it in there as well. So 2.5 times 44 gives us 110. So the length from X to Y is 110 micrometers. So we calibrated our eyepiece graticule, and then we used that calibration in a calculation, all based on image work. So there's no need for you to have a slide to do this. So there's very valuable resources in these microscope questions in your prac papers. So if we move on, we have another image. See, image work is fantastic. In fact, this is the end of the question. So what have we got? Figure 2.5 is a photomicrograph of a transverse section of the same leaf as in figure 2.4. Now you can see the whole triangular beauty of this leaf. You can see that we've only got one vascular bundle in the middle, and we've got all sorts of fun games going around on the outside. So annotate figure 2.5 to describe two observable differences between the leaf in figure 2.5 and the leaf on J1. Draw label lines to two different features and use only the labels P and Q. Next to each letter, describe how each feature on the leaf in figure 2.5 differs from the leaf on J1. So obviously we don't have J1, but this is different to the leaf in our textbook, so we can compare it there. So for example, one that leaps out is, this is a triangular shaped leaf, and the leaf in the textbook is a classic midrib and blade shape of leaf. So we could give we want P, okay? Another one, remember when we did that plan diagram, there was more than one vascular bundle, whereas if you look at this leaf, there is only one vascular bundle. So there would be Q. We also have two layers of epidermis here, and we only have one layer of epidermis in the textbook. So there's lots of different things that we can pick from, and you got to then pick the two that you want to work with, or the two sort of obvious and most crucial ones looking at functionality of these two leaves. So next to each leaf, describe how each feature on the leaf in 2.5 is different from the leaf on J1. So here, Q would be only one vascular bundle in leaf. Okay, so the converse of that is more than one vascular bundle. Okay, but we want to describe the features of this leaf. That's what the focus of this question is. And then for P, leaf is triangular in cross-section without defined blade. Okay, so it almost looks like wine grape big midrib rather than midrib and blade, like the classical leaf in your textbook. Okay, so you only use the labels P and Q, and then you describe what is special about this leaf. So this is an alternative to doing one of your comparative tables. And they want to know, can you follow instructions? Can you do simple P and Q, and can you also then annotate? An annotation, remember, is an expanded label. It's giving a description rather than just giving a single label. And that brings us to the end of the question 2. So you might find this in your pracs as well, that the microscope work actually takes you less time than the hour, whereas the wet prac work might take you more time than the hour. So it's really important that you use your time wisely. So if you do your microscope work first, then you start reading what's going on in the experiment. Can I draw my graph already? And if you're doing your experiment first, don't get yourself in such a frazzle if you don't quite finish, because you can finish off things like drawing the graph when you're sitting by your microscope. Okay, so you've got 1 hour on each station, but you can work throughout the entire paper at any time within the 2 hours. So use your time wisely.